Okay, so this is question 1 from the 2023 Higher Human Biology past paper. The diagram represents the structure of a section of DNA. So this is a section of your DNA. It's not been given in the double helix form. It's been given in a slightly different way. And our job is to name the repeating units that make up DNA. Well, we know that DNA is made up of something called nucleotides. So nucleotides are the building blocks of DNA. So all DNA is made up of a nucleotide, and these nucleotides have different parts to them. They have the base, which is represented by the letter, and then these two parts, which we'll talk about in the next part of the question. So B, name the parts of these units joined by strong bonds that make up the backbone of DNA. Now the backbone word here is something that I draw my eye to a little bit, because we always speak about something when we're talking about DNA called the sugar phosphate backbone. Now those are the two words I was going to mention earlier on when we're talking about the nucleotide bases. So this little circle here, this is our phosphate unit, and the pentagon here would be our sugar. If you want to be extra brainy, you could say it's a deoxyribose sugar. It's the name of the sugar there. But for this mark, saying sugar and phosphate is totally fine. That would absolutely get you the mark. Our next job is to identify one letter from the diagram that labels the 3' end of a DNA strand. So whenever we're given DNA, it's always important that we can understand which side is 3', 5'. So the side with the phosphate sticking up, that is going to be 5'. Okay, so on this side, because DNA runs anti-parallel, the 5' end is going to be down here, because this is the direction that the phosphate molecule is going in, if that makes sense. So that means then that the other side there is going to be the 3'. So let's just draw that in. That means that this side here is going to be 3', which means this side here will also be 3'. So if we're trying to figure out what letter labels that 3' end, then the two letters we could possibly write are going to be X, or we could write Y, either or to get that mark. So we're going to continue on. Question B. The first stage of the production of a protein involves the synthesis of mRNA from a section of DNA. Name this stage. So this is all about production of a protein. And because we're talking about synthesizing mRNA, that's going to be the first part of our two-step process, okay? So we have the process of protein production is transcription and translation. So transcription is the production of that mRNA molecule. Translation is turning that mRNA molecule and using that to translate it into a protein. So the first stage here is going to be transcription. Describe how alternative RNA splicing can produce different proteins from one gene. Now, this is a really good question. We're going to look at a diagram to help us understand this. On the left-hand side, we have RNA splicing, which is basically how we go from a section of DNA into our pre-mRNA or primary mRNA, and that's basically our mRNA transcript that still has our exons and our introns. Remember, exons are our coding regions, introns are our non-coding regions, and it's basically all about how we turn the sequence of this mRNA into a mature mRNA transcript in which we've removed the introns and we're just left with those coding regions. Alternative splicing is a similar idea. We have our DNA section, our primary mRNA section, but instead of using the same order of our exons, we switch up the order of the exons. So you can see here, we have exon number 1, 3, 5, exon 2, 3, 4, 1, 2, 5, whatever it is. And the reason we do this is because if you splice your RNA in this way and mix up the exons, you have a greater diversity of the type of proteins and the type of mature mRNA transcripts you can produce. So because you can make more mRNA transcripts, you can make more diverse proteins. So let's think about how we're going to translate that into our answer. So in the mark scheme for this question, you could have said different mature mRNA transcripts can be produced, or you could have said different exons are removed or retained, and it's all coming from this idea that it's from the same section of DNA. And the final question here, describe the function of tRNA in protein synthesis. So let's remind ourselves on the tRNA molecule. So the tRNA molecule looks a bit like this. It's a funny looking molecule. tRNA stands for transfer RNA. It's made of RNA the same way mRNA and rRNA are made of RNA. It has an amino acid binding site on one side and it has an anticodon on the other. Now, these molecules live in the cytoplasm, and what their job is to do is that when the mRNA transcript leaves the nucleus and goes to the ribosome, the tRNA anticodon will pair up with the codons on the mRNA and bring amino acids to start growing that protein. As we know, proteins are made of amino acids. So the tRNA's job is to bring those amino acids to help start growing that protein and producing what is going to be our final protein here. So we've recapped a bit about tRNA as a molecule, talked a bit about translation. Let's think about what we could say to give a possible answer here. The mark scheme gives three possible kind of concepts they want you to touch on in order to get those 2 marks. You don't have to mention all 3, it's any 2 of those 3 concepts. The first concept is that tRNA carries amino acids to the mRNA transcript that is going to be at the ribosome. So you get a mark for describing the fact that tRNA carries amino acids. You'll get a mark for saying that tRNA has an anticodon that is complementary to the codon on the mRNA. And you'll get your third mark there will be for saying that tRNA detaches from the ribosome and can then go and collect other amino acids, or basically that it's reusable in this process, and that it will ensure that amino acids are in the right order, because the anticodon is going to be specific to the codons, and that's going to make sure amino acids are in the correct order. So those kind of ideas are what they're looking for in order to get those 2 marks. Again, they're all attached to the idea of the tRNA molecule itself and its anticodon, and it's carrying amino acids, and then what it does at the ribosome with that mRNA.